All right, thank you very much for that introduction. So yeah, this is a talk in protein mass spectrometry, which may not be as popular a field as, say, for example, single-cell genomics these days. So I'm going to start out with just a one-slide background about what data is before diving into the project. But I also, if I'll try to give background as I go, but I'd like to, invite people to interject with questions throughout, mostly clarifying questions and that kind of thing. If you have like a big conceptual question, we may postpone it till the end. But I'm happy to get interruptions as I go, and I can adjust as needed. So just to kick us off, so we're all on the same page, I want to introduce just the data type that we're going to be talking about for both of the projects that I'll describe today. So this is Tandem mass spectrometry data, and it's typically used for identifying, and I should say, and quantifying on this slide, proteins in complex mixtures, whatever kind of tissue sample or organ sample that you have. So obviously proteins themselves are very complicated. They're difficult to deal with biochemically. And so the way tandem mass spectrometry works is that you begin by digesting the proteins down into subsequences called peptides. And then those peptides are put through a tandem mass spectrometer, which is itself complicated, but I'm just going to summarize it with a picture. It's a complicated device that separates all the peptides using liquid chromatography and then measures them twice in two different ways. But the upshot is that this device produces, on the order of 10 or 20 or 30 mass spectra per second. And a typical run will take 30 minutes to, an hour or an hour and a half. And these are the primary data objects. Nominally, each mass spectrum, if things go as you hope, corresponds to one peptide species. And then the idea is you use some kind of computational analysis to try to infer what was the peptide sequence responsible for generating that spectrum. There's a whole other part of the analysis which involves then quantifying how many copies of that peptide you expect to be in the mixture. But we're going to focus today just on the identification, or the detection of the peptides in the mixture. So that's the primary data. And the first project I'm going to talk about is one that started a while ago when Damon May was a graduate student in my lab. And then when he graduated, Wout Bitremier took over as a postdoc in the lab and took the project to fruition. And it was recently, it just showed up in Nature Methods, I think a month ago or a month and a half ago. And it was all done in collaboration with Jeff Bilms, who's a machine learning expert in electrical engineering department here at the UW. So the motivation for this project has to do, well, one motivation anyway, has to do with the idea that we'd like data to be reused more frequently. So, it is certainly the norm in proteomics, just like in genomics, for example, that if you publish an analysis, that you're required to make your data publicly available as well. And so there are various kinds of repositories where you can deposit your data. And typically the way this works is you have this kind of workflow where you generate your data, you analyze it, you write your paper, and then you're like, oh, in order to submit this paper, I have to deposit my data. So I send it off and I put the URL in my paper and, all's good. And then I start my next project. We would like instead to encourage people to deposit their data earlier so that they could then use other data to try to make sense of the data they have in hand. The idea is if there's, thousands and thousands of experiments out there in these repositories, they should be able to inform us about the data that we just collected. So the particular hypothesis that we wanted to pursue here was that we can use supervised machine learning to try to learn an embedding of peptide or of spectra into a latent space that can help us to assign peptide sequences to spectra when we get a new set of spectra coming along. So that idea can be instantiated in this idea that we're going to take each mass spectrum, which is a very complicated kind of data and typically requires specialized algorithms and actually is kind of has a larger memory footprint if you have billions of these things, and compress each spectrum into a fixed length vector, an embedding, that is simple, and then you can apply out-of-the-box algorithms to make sense of these mass spectra. So conceptually, you can think of this like we're going to try to embed all these spectra into a latent space. So this would be a two-dimensional space, but obviously in reality, it would be higher dimensional, in such a way that if two spectra have the same generating peptide, then they're likely to be close together in the space. And then the idea is when we come along with new data, we just embed it into this space and look for the neighbors, and they can tell us what was likely to be the peptide that generated. So to do this learning, we first had to deal with the challenge that the spectrum itself is a complicated data type. So we had to represent things like what parameters of the experiment that generated the peptide, like what was the mass accuracy of two different properties of the spectrum. And then information about the intact peptide that the mass spectrometer collects, like what's the total mass, what's the total charge, and then of course the ratio between them, which is actually what is directly measured. And then the main thing is all the fragment information. When I draw a spectrum like this, the x-axis of the spectrum is the mass to charge ratio, and the vertical axis, the y-axis, is an intensity or abundance that sort of reflects how frequently each m over z over mass to charge value was observed in the mass spectrum, or how much intensity was observed. And so nominally, each of these peaks corresponds to a different fragment of the peptide. And so you have to figure out some way to represent all that fragment information. We take all of these three different representations of the data. So there's 61 features that represent the m over z, the mass, and the charge. There's 2,449 features that represent the fragment values. And then we also have this kind of what's called an empirical kernel map. So you have 500 reference spectra features, which are sort of a trick to capture the fact that the mass to charge ratio of the m to z, the measurement of the m to z axis is extremely precise. And when we do this, these binned intensities over on the left over here, we lose some of that precision. And so the reference spectra features are similarities to a randomly selected set of reference spectra that are computed in high resolution, with the concept being that that high resolution information can then get reflected in these additional 500 features. So then the embedding mechanism is relatively straightforward in a deep learning setting. You have some CNN blocks, you have some max pooling layers for the fragment features, and then you have the dense layers for these other features, and they all get combined. And the embedding dimension that we selected was 32. This was sort of this is of course a key hyperparameter. We selected 32 for sort of two competing reasons. 32 was small enough that it was going to be useful for being able to embed tons and tons of spectra and still manage them efficiently. As you'll see later, we're doing things like clustering hundreds of millions of spectra, and every time you add more dimensions, you add more memory requirements and more time to the downstream algorithms. So that's an important feature, but of course you want it to be enough dimensions to capture most of the information in the spectrum. So that's how you embed one spectrum, but in practice, what we're interested in is learning about pairwise relationships between spectra. So we do this with a fairly standard approach, which is the Siamese architecture. You have two copies of the embedding network. These vertical purple bars here represent the fact that the parameters of these two networks are tied. You input two different spectra, and then you have a label that says the label is positive if the two spectra were generated by the same peptide, and it's negative if they're generated by different peptides. The particular loss function that we use is a contrastive loss function that looks like this. So you have your two spectra, and then the weights and the labels. And the labels, as I said, are 1 or 0 for whether the same peptide or different peptide. And then you take the embedded spectrum, both of the embedded spectra, and you basically are trying to make sure that all the ones that are labeled similarly are close together, and the ones that are labeled differently are far apart, subject to some margin. That's the M is the margin. So the actual software, the algorithm, is a recursive acronym. Gleams is a learned embedding for annotating mass spectra. And so we call this a Gleams embedding. And we ended up training this from an existing repository. So Massive is the name of one of the public repositories for mass spectrometry data. It contains hundreds of millions of spectra, but they, several years ago, did a systematic annotation of most of the data in the repository. And they selected a subset of spectra that they could identify with high confidence. So that consists of about 30 million, what we call PSMs, which stands for peptide spectrum match, that have an estimated false discovery rate of 0.1%. And then, of course, we split these into training, validation, and test in such a way that you don't have the same peptide occurring in different sets. And so then in the end, you really have 540 million positive pairs and 11 billion negative pairs for training the Siamese network. So once we do this, we can do things like, so we've got a 32-dimensional embedding. We can then project it down to 2D using any kind of dimensionality reduction procedure. This one is a UMAP. I should have put the citation for UMAP on the slide, but anyway, they're UMAP projections. And these have been colored in two different ways. On the left, we're coloring by the precursor charge. So the charge of the peptide that ranges from 1, in practice is mostly to 5. There's very few that are 6, 7, or 8. And then also the precursor mass over charge. And what you can see is a lot of the structure in this learned embedding is reflected in these two main parameters, which is not super surprising because these are sort of major parameters of the space. One of the things we wanted to check just to make sure this is working is it the case that if they're close together, they're associated with the same peptide sequence? And so you can make a plot that shows for the labeled test set, what proportion of spectra, pairs of spectra, have the same peptide sequence as a function of distance in the embedded space. And so you can see that for very close regions, there is quite a bit of similarity here. And then if the second line here is considering the localization of post-translational modifications. So that's something I haven't really touched on yet, but the idea is some peptide sequences have additional molecular groups can attach to them, like a phosphorylation or an oxidation, for example. And if you have, for example, a methionine oxidation, but your sequence contains 2 methionines, it's very hard to tell the difference between that same peptide sequence with an oxidation on 1 methionine versus the sequence with oxidation on a different methionine. And so if we treat those two as the same, then you can see that the curve gets a lot better in the sense that you have a higher percentage. So many of the confusions have to do with this very difficult to discriminate localization of PTMs. And then we can also use this kind of information to set a threshold to decide, okay, if we wanted to make an annotation and we have about a 1% false discovery rate, we can figure out exactly what that threshold would be. Okay, so the question is, what do we do with this? Now that we've embedded all my spectra, I've got this 32-dimensional space, what good is it? The main thing that we ended up doing was applying an existing clustering algorithm to the data. We actually, in the earlier version of this work, experimented with, sort of making up our own clustering algorithm, but then finally realized that that's not really the point. We're supposed to, since we have these nice vectors, we should just be able to drop them into a clustering algorithm. And in the work, actually, in the paper, we compare like 4, I think, different clustering algorithms. And the one that gives the best performance is this DB-scan algorithm that I'm sure many of you are familiar with, that Martin Nestor published back in 1996. I'm not going to go into how DB-scan works. This is just the figure from the Wikipedia page about it. But the point is, it's an off-the-shelf clustering algorithm that scales very well to very large data sets. So here is a comparison. Now we're comparing Gleams, which is this supervised sort of embedding approach, to 3 existing algorithms that are unsupervised clustering approaches, all on the same test set. And what we're showing is the number of incorrectly clustered spectra as a function of the number of clustered spectra. And you make these curves by varying various parameters of the clustering algorithm. So the clustering algorithms usually have something that controls how many clusters the algorithm produces. And then for Gleams, we can vary the threshold. And what you can see is that Gleams is doing well, but it's doing particularly well at low error rates, which is where we would argue that that's the most interest lies in getting below 1% error rate. Furthermore, you can look at other things, like, for instance, completeness. This is a standard metric for evaluating clustering algorithms, which means you're essentially asking how often are clusters or spectra with the same label clustered together. So if you have an example on the left, both of these clusters are correct, but it's incorrect in the sense that you've taken all 4 of peptide A and split them into 2 clusters, and it's clearly better to have a more complete clustering like the one on the right. And in that regard, Gleams does very well. So we're getting clusters they're basically larger clusters that gather more spectra together, and we do better than all of the other methods across a range of percentages of incorrectly clustered spectra. This is also reflected in the fact that we just produce larger clusters on average. So this is kind of the same qualitatively similar data, but now we're actually looking at the cluster size, sort of a CDF of cluster size. And again, Gleams has larger clusters. And then I think more importantly, we showed that Gleams produces clusters with data drawn from more diverse studies. So often in these kinds of studies, you get batch effects where spectra from one study resemble one another, but don't resemble so much spectra from a different study. And the supervision can help overcome that. And so this is just showing clusters that have data from different original experiments. And so we have a large proportion of clusters that have, say, for example, data from 15 different experiments. And that's rare for some of the other clustering algorithms. The last thing we did with Gleams was to explore the so-called dark proteome. So this is a concept in proteomics where you have spectra that you keep seeing over and over again, and no one has ever successfully been able to identify them. So they're observed across many experiments, but consistently not identified. So we took all of, a large collection of data from Massive, and we looked at the ones, of course, there's the ones in Massive KB that were successfully identified in the original analysis, and then a whole bunch more that they did not identify. And then we applied Gleams clustering, and we did 2 things. First, we just propagated identifications within clusters. So if you have a majority of spectra in a cluster that are labeled with a particular peptide, you can annotate the rest of the spectra in that cluster. And then we took the medoids of each of the remaining unidentified cluster and did a more expensive search operation called open modification searching, which would be computationally infeasible to do systematically across the entire data set, but you can afford to do if you have Gleams telling you which ones are interesting and worth expending the compute on. And then we identify additional ones and propagate their identifications within clusters as well. And so when we do that, we're able to actually identify 71% more spectra. So that's the first project, Gleams. Gleams is now, as I mentioned, it's a published tool, it's a learned embedding, and the goal is to enable efficient repository scale data analysis. One of the things that we're exploring right now is what other uses can we put to these compact representations of mass spectra. So you can imagine using them for example, facilitating, speeding up various kinds of analyses. And we're hoping that other people will begin to use them as well as a way to facilitate other kinds of applications and particularly transfer learning applications and machine learning. The second project I'm going to tell you about is work that was done by a graduate student, Mele Yilmaz, in my group, and in collaboration with Siwung Oh, who's a machine learning expert in the computer science department. And then it's also done with two former postdocs in my lab, Will Fondry and Wout, the one who did the Gleams work before. And an initial version of this appeared in ICML last month, or maybe in July. And we're working on a full version to be submitted to a journal, hopefully within the next month. And the topic we're talking about here is another alternative way to assign peptides to spectra. And it's called de novo mass spectrometry sequencing. So this is, I alluded to this before, but there's basically two different ways to assign a spectrum or a peptide to an observed spectrum. And they're referred to as de novo sequencing and database search. So in the database search procedure, you would basically take your experimental spectrum and systematically compare that spectrum to theoretical spectra that are generated from a sequence database. For example, all of the proteins in the human proteome, for example. And you compute some kind of score and you report for each spectrum what's the best matching peptide sequence from the database. The other alternative is to do this kind of analysis, but get rid of the protein sequence database. So in that sense, you're making the hypothesis space a lot larger. It could be any peptide in the universe and not just peptides that occur in the human proteome. And so it makes the problem harder, but it also gives you more flexibility to identify things that maybe you didn't know about before. So this is called de novo peptide sequencing. And of course, the drawbacks to database search, I think, should be fairly obvious. So you have to know a priori which peptides are likely to occur in the sample. So if you're doing this in a model organism, if you're working in human or mouse or something like that, we have the complete genome. We can typically infer what all the peptide sequences would be that would be produced by that genome. The drawback, of course, is that you typically don't capture things like genetic variation. You might not get all the post-trans, that's supposed to say translational modifications, PTMs. And there's also some issues around handling non-standard enzymatic digestion, different ways of digesting the peptides initially. But there are other cases where it gets more difficult. So immunopeptidomics is an example where the peptides are produced by the cell and not through digestion. And so they don't follow enzymatic digestion rules, which means the space of possible peptides gets really large and your database can get unwieldy. And then even more difficult is something like metaproteomics, where you're doing like ocean samples or soil samples or even the human microbiome, where there are organisms in there that no one's ever sequenced before. And then we just don't have a full, complete database. And so in that case, you kind of have to do de novo if you want to try to identify the sequences that aren't in your database. So the idea is we're going to take as input to our model an observed mass spectrum. Again, you have the mass to charge ratio of the fragments here and the intensities on the vertical axis. You also get the mass and the charge of the intact peptide because that's observed by the mass spectrometer. And then the goal is to output the peptide that was responsible for generating that spectrum. It might seem like this is a relatively straightforward thing. So just to make it clear, one of the things that makes it difficult is that if you have an observed peptide, it's fragmenting in the mass spectrometer at any particular location along the peptide backbone. And each of those produces two ions, a prefix ion and a suffix ion, which in the field are known as B ions and Y ions. And all of those are observed in the mass spectrometer, but of course, they're not labeled as B or Y. We don't know which ones are prefixes and which are suffixes. And so you have to infer that from, for example, the observed mass differences between pairs of peaks would correspond to, for example, the mass of a particular amino acid. But the problem is sometimes these fragmentation events don't occur, so some of these peaks are missing. Sometimes there are unexpected fragmentation events where it fragments at different locations along the backbone, or the peptide can fragment but then lose an additional molecular group, like a water group or ammonia group, and then you get a slightly different molecular mass for the fragment. And then you can also have co-eluting peptides that contribute additional peaks that can distract you, and of course, just noise in the data. So all of that makes sequencing one of these real spectra challenging. So we're not obviously the first to have done this. People started developing de novo sequencing algorithms back in the 90s. And for a while, most of the approaches used either heuristic search procedures, just kind of cobbling together using some simple rules what the sequence might be, or for a while the dominating approach was a dynamic programming procedure where you build up something called a spectrum graph and look for pairs of edges that have the expected differences. But for the last 7 years or so, the state of the art has been machine learning, and in particular deep learning. So these are some of the existing methods with the year that they were produced. And the first machine learning approach to this problem was in 2015 from this algorithm called novo. And then starting in 2017, the state of the art was deep novo with several other competing methods that have been published since then. So even deep learning methods, though, haven't solved the problem perfectly. So one thing is the accuracy still leaves room for improvement. You're correctly typically assigning only about 40 to 60% of the spectra they're generating peptide. And then they tend to be fairly complicated models combining multiple neural networks along with post-processing steps to sort of clean things up. And then most of them also require this discretization of the MZ axis that I've mentioned before, which is giving you a trade-off between capturing the actual precision of the instrument and then also having lots of parameters in your model if you use very fine scale binning. So just to push on that a little bit more, this is what's typically done. Most times you use a machine learning method, you have to have a vector representation of an input, but actually a spectrum is not a vector, it's a bag of peaks, where each peak is a tuple of an MZ and an intensity. And so typically what happens is people discretize the MZ, and then the entries in the vector become the intensities. But when you do that, you introduce not only sparsity if you use very fine bins, but also edge effects. So for example, if you have these MZ values and these intensities, you could bin at these large MZ windows, and then you would report, say, the maximum intensity, or you could use the sum of intensities within that window. You get 75 here and 100 here and 40 here, or you can use finer bins. But as you use finer and finer bins, you start getting more bins with zeros in them. And in practice, we really need something like on the order of 0.01 MZ to get close to the precision of the instrument, because it's very precise. And then you have vectors of like 200,000, which is challenging for training, loading onto your GPU and training efficiently. And then also, of course, as you get those very small bins, you start introducing edge effects where two peaks that happen to have an edge between them get separated, even though they actually have very close MZ values. So we decided we'd like to think of this instead of a sort of vector representation, think of this as a translation task, where you can think of I said a spectrum is really a bag of peaks, but if you order those peaks by their MZ, then you can think of it as an ordered series of peaks. And of course, a peptide is an ordered series of amino acids. And so the idea was there's all these models from NLP, natural language processing, for doing translation between sequences, and the state of the art these days seems to be to use transformer models. And so we thought maybe we can use a transformer model to carry out this translation task. One of the key things about a transformer model is that it has this notion of self-attention. Those of you who are familiar with these models have heard a lot about this recently, but the idea is that you have a function in the model. Essentially, it's learning a set of parameters that tells you connections between elements in the input and learns those dynamically based on the data. So you have a query, that says, if I focus on this particular item in this sequence, then I'm asking how much does its meaning depend on each of the other items? And there's also an items representation without additional context. So if you have, for instance, this sentence, you might have attention connections between the tokens in the sentence that represent these learned relationships between pairs of elements in the input sequence. So in our setting, we're thinking of peaks as words and spectra as sentences. And so rather than learning these kind of relationships relating words to other words in the sentence, we're learning relationships between pairs of peaks. And those might be, for example, these two peaks seem to be related to each other because their difference is equal to the difference between, is equal to the mass of a known amino acid. Or maybe the difference between these two corresponds to, or the relationship between these two M over Z represents the complement ions. So this would be the prefix mass, and this other one is the suffix mass. And the idea is that the attention mechanism might enable the model to pick up on those kinds of pairwise relationships. So the idea is we have these existing methods, deep novo, SMS, and point novo. They have all these properties that they're using, for instance, convolutional neural networks or recurrent neural networks, this existing point net approach for handling the MZ discretization. And Casanovo's difference is twofold. One, the idea of using a transformer, which is itself new. And then also some of these methods, most of these methods involve some complicated post-processing that involves a dynamic programming procedure to try to enforce that the mass of the inferred peptide is very close to the mass, the observed mass of the peptide. And we found that it was simpler just to basically put on a filter, so not do this more computationally and complicated dynamic programming step, and just basically say, if the prediction fails to obey this relationship, then we're just going to make no prediction at all. And it turns out to work pretty well. So here's just an overview of the model architecture. You've got basically two, the transformer encoder and the decoder. So the encoder is taking the spectrum, and the decoder during training is taking the peptide, but of course at test time it's just outputting the peptide sequence. And so you have individual peak encoders and encoders for the individual positions, and then those go through the main transformer architecture. And then, as I said, we filter out the implausible predictions in a post-processing step by comparing the observed and predicted precursor mass after the inference. Okay, so this is fairly standard, but just to make sure we're all on the same page, when we're doing decoding in this model, the way that's done is different during training and versus during inference. So during training, at each decoding step during training, the decoder is fed the preceding amino acids in the original peptide. So it's basically, it's called teacher forcing. You're forcing it to predict the correct amino acids before your current position, and then being asked to predict the next one, and that's where you get your gradients from. And the decoding stops when the original peptide link is reached. During inference, of course, you don't have the original peptide sequence, and so you only get the previous amino acid predictions, not the true previous amino acids. And then you stop when your predicted mass exceeds the observed predicted mass, or the observed precursor mass. So the loss is just a mean squared error kind of loss. We can represent the original peptide sequence as a matrix. It's kind of like a one-hot encoding, where each column is a position, and or actually each row is a position and each column is an amino acid. These are binary, and then the predicted peptide sequence is output as real numbers, and we get a mean squared error between the top matrix and the bottom matrix, and that becomes the loss. We used this existing benchmark. It was originally from that deep novo paper in 2017. It's a 9-species benchmark. You have about 1.5 million mass spectra, and the idea is we're cross-validating by species. So we would train on 8 of the 9 species and then make predictions on the 9th one. And it's not a perfect separation of train and test set in the sense that some species do share a few peptides in common, but it's a very small percentage of shared peptides. So it's mostly distinct peptide sequences. And you can see that the number of peptide spectrum matches varies by species. And we have quite a range of species, all the way from human down to, there's mouse here, and then bacillus and so on. And then I mentioned this already, we have training on 8 species, testing on the left out species. And so you have about 100,000 spectra per test set. The performance measures that we use are fairly standard. There is some discrepancy about whether people use something called precision and recall or precision and coverage. This isn't really a binary classification task, so recall isn't formally definable in this setting. So instead we use precision and coverage. So precision is just what you would normally expect. It's the number of correctly predicted peptides over the total number of predicted peptides. And then coverage is just what proportion of the spectra have we made any prediction for. So we can trace out a precision coverage curve by lowering our confidence threshold, the score that the method outputs, and making more and more predictions. So here's an example of a precision coverage curve. And we're comparing in this one, Casanovo to deep novo, which was the previous best performing method. And what you can see is, obviously precision starts out very high and then drops. This shows that after we've made predictions on 100% of the spectra, then you're getting about a little bit below 50% of them getting correctly predicted. The point is that for varying levels of coverage, we're still consistently beating deep novo. Right here, the star is the location where we actually stopped making predictions. So after this, everything is incorrect because we just said, we don't know what these ones are. And so these are the ones that are the actual real predictions up to here. In this particular case, so we can measure the area under the curve. In this particular one, we get 0.78 versus 0.585 for deep novo. If we look at this for all of the species, you do see a consistent trend that we have a mean improvement of the area under the curve of 0.13. And even in the worst case, which seems to be tomato, we're still getting a significant boost here. We also can measure performance at the level of amino acids. So you can ask for each peptide sequence, you can ask how well do we predict what is the amino acid at this position. So in that sense, you can say, for instance, if we predicted almost the correct peptide, but we swapped the position of 2 amino acids, then you can get it sort of 80% correct or something like that. And so you can also make an amino acid precision curve. And for this one, we also improve upon deep novo, although the improvement is not quite as pronounced. You can see that for the red stars are all above the line. And then beyond that, I think in this part of the curve, sometimes we don't do as well, but I would argue that that's not the most interesting part of the curve anyway. So what we're subsequently doing, so in the initial paper, we just used a greedy decoder. We've subsequently worked on a better decoding scheme, which is a standard decoding scheme called beam search, where in the greedy decoding, you predict the amino acid with the highest score, whereas in beam search, you consider sort of the n highest scoring amino acids in each position, and then assign the highest scoring peptide with a matching mass to each spectrum. So you kind of have a tree during the decoding process. And as I said, this isn't a novel thing, it's a standard kind of decoding to do, but it does seem to make a significant difference. So this just shows a difference between greedy decoding and beam decoding. And so we're implementing that in Casanovo in the published version, or the released version of Casanovo right now. Bella's working on that right now. And there is lots of other stuff that we're working on for the final paper, which will involve more applications. So showing that you can use Casanovo, for example, to revisit that dark proteome for doing metaproteomics analysis and for doing immunopeptidomics analysis. So all of those things are coming soon, and we seem to have some at least promising results on several of those fronts already. Both of these sets of code are publicly available, open source. You can get Gleams from this GitHub URL here. You can get Casanovo from our lab's GitHub here. And I think that's it, and I will be happy to take questions.